So today is the last lecture. The subject of our lecture today is the urinary system and genital systems, reproductive systems of the human body. Today we will talk about the urinary system and its development, general morphofunctional features, hormonal regulation. And then we are going to talk about the histophysiology of urine formation, hematourinary and hematofollicular and hematotesticular barriers. So the urinary system, female reproductive system, and the male reproductive system, they are the main topics of our lecture today. First of all, let's talk about the urinary system. We know that urinary system, it eliminates the toxins and the excess of fluid from the body, and at the same time it preserves necessary ions, salts, water inside of the body. At the same time, the urinary system provides production of some hormone-like substances like renin, erythropoietin, and also produces some prostaglandins. Kidney also help in the production of the active form of the vitamin D. So let's talk about the structure of the urinary system. The two kidneys, two ureters, urinary bladder, and the urethra together form our urinary system. The structure of the kidneys, we are looking here, and we can see that each kidney, it has bean shape, it has convex and concave sides. In the concave side, we have hilum of the kidney through which arteries, renal arteries and nerves enter the organ, and renal veins, ureter, and veins, they exit the organ. The organ is covered by connective tissue capsule. Under of it, we have the cortex and the medulla. The medulla include the pyramids. Approximately in each kidney, we have approximately 12. It may be 18 pyramids. Pyramids compose the medulla. Actually, the subdivision of the cortex and the medulla, that they are not so abrupt as in some regions. The cortex continue into the medulla and it forms renal columns. As we see here, renal columns, they are located between the pyramids and they separate pyramids from each other. Also, we have the cortex inside of which we have continuation of the medulla into the cortex. And the continuation of the medulla into the cortex, it will called medullary rays. Medullary rays, actually these are continuations of the tissue of the medulla into the cortex. Each pyramid has a base which faces the cortex, and the apex which faces the lumen of the organ. Each apex of the pyramid is surrounded by the minor calyces. 2 to 3 minor calyces open into the major calyces, and the major calyces, these are small spaces which together will open into the large one lumen of the organ, which is called renal pelvis. From the renal pelvis arise ureter. In the slide of the kidney, we can distinguish that the cortex, it includes these nodular structures, rounded structures, which are together with the tubules. They form a cortical labyrinth, or they called uriniferous tubules. And between the tubules and these corpuscles, there are very little connective tissue, approximately 7% of the cortex, and just 30% of the medulla include connective tissue. It is the interstitial tissue which is located between the tubules. In the center of, as we see here, in the center of this slide of the cortex of the kidney, we can see medullary rays, and around we have these tubules. Now we will understand what does it mean, this structure, which include medullary ray in the center and around these corpuscles. First of all, we have to understand that the structural and functional unit of the kidney, these are the nephrons. Nephrons, these are functional units of the kidneys also. There are approximately 85% of all nephrons, these are cortical nephrons, and only 15% of them they called just juxtamedullary nephrons. Each nephron include, first of all, it starts with a renal corpuscle, which have rounded shape. Then from it arises proximal convoluted tubule. As we see here, they are long and folded, these convoluted tubules. Proximal convoluted tubules, they continue into the proximal straight tubules. Proximal straight tubule descend towards the medulla, and here it continue into the thin tubule. Thin tubules have descending limb, handless loop, it forms hairpin loop, and then it has ascending limb. Then ascending limb of the thin tubule continue into the distal straight tubule, which continue into the distal convoluted tubule. And distal convoluted tubule, which is written here, we can see the name here, distal convoluted tubule. It opens into the collecting tubules. So at the distal convoluted tubule, the nephron finishes. The parts of the nephron finish. So all of the structures which we mentioned just now, they are parts of nephrons. That is a cortical nephron, which main parts, whose main part located inside of the cortex. Just handless loop is located at the superficial layers of the medulla. As we see here, that is a medulla and that is a cortex. But in juxtamedullary nephrons, their main parts, the proximal tubules, distal tubules, and corpuscles, they are located at the junction between the cortex and medulla, and their straight tubules, their proximal straight, distal straight, and all of the thin tubules, all parts of the thin tubules, they are located inside of the medulla. The distal tubules of nephrons open into the collecting tubules. Collecting tubules, they join each other like branches of the tree, and they form the larger collecting tubules, which descend towards the medulla. And consequently, with descendance of the collecting tubules, their diameter increases, and finally they continue into the large collecting tubules or collecting ducts, which are called Bellini's ducts, and they will open at the apex of the pyramid. At the apex of the pyramid, there are so all of the collecting tubules, collecting ducts will open at the apex. That's why the apex of the pyramid has lots of openings, and it has sleeve-like structure. That's why it's called area cribrosa. It means that so much openings are present there. So the area cribrosa is located at the apex of the pyramid, and here it is surrounded by minor calyx. Minor calyx, as we know, continue into the major, and it continues into the renal pelvis. So the urine which is produced inside of the nephrons will flow into the collecting tubules, and through them it will flow into the lumen of the kidney, and through the ureters it will be delivered into the urinary bladder, which is the next part of the urinary system. What is a renal corpuscle actually? As we see here, the renal corpuscle, it includes glomerulus, which is the network of capillaries, and it is surrounded by Bowman's membrane. It is a Bowman's capsule. The Bowman's capsule, it surrounds the corpuscle, it surrounds the glomerulus. And it is here will start the production of the urine. We also have to know that all of the kidney we can subdivide into the lobes. Each lobe of the kidney, it includes one pyramid. As we see here, the pyramids are in medulla, and in this picture we can distinguish 3 pyramids. The apex open into the calyx, as we know, and the base attached to the cortex. So one pyramid which together with the associated part of the cortex, they form one kidney lobe. According to number of pyramids, we have number of lobes inside of each kidney. The kidneys also include lobules. So what are the lobules actually? The lobules are cortical structures, and they present just inside of the cortex, and they are associated parts of the pyramid, which are associated with each pyramid. In each lobe, inside of each lobe, we have lots of lobules. The center of each lobule is formed by the medullary ray, as we see here, medullary rays. And inside of the center of the medullary ray, we have collecting tubule, as we see here. That is a collecting tubule here in the histological slide, which is stained by the hematoxylin and eosin. So all of the surrounding nephrons via their distal tubules open into that particular collecting tubule. And these together, the medullary ray, which include collecting tubule in the center, there are also other tubules, but the main principal element here, that is a collecting tubule. And together with all surrounding nephrons, which open via their distal tubules into this collecting tubule, together they form one lobule of the kidney. One lobule of the kidney. So that is a general plan of the structure. Now we will talk about, and in this picture also we can see renal columns which separate neighbor pyramids from each other. As we know, they are the continuations of the cortex into the medulla. Then what is the blood supply of the kidney? We know that the kidneys, they receive the blood from the renal artery, that is a branch of the abdominal aorta. During each minute through our kidneys, there will be pass approximately 1,250 milliliters of blood. It means that during 5 minutes, all of the blood of the body can pass through the kidneys. So the kidneys has very rich blood supply. The order of branching of the arteries inside of the kidneys, it may be represented as the renal artery, which enters the organ. It subdivides at the hilum. It subdivides into anterior and posterior branches. Then anterior will supply the anterior part, and posterior one will supply the posterior part, the back of the kidney. Then these arteries subdivide into segmental arteries, each of which supply each particular segment of the kidney, and then each segment is supplied by just only its own segmental artery, And we have to remember that there are no any collaterals between the branches of the neighbor segmental arteries. It means that if in one particular segmental artery will be, if one of them will be clogged, if there will be some problem, the neighbor segmental artery will not be able to supply that particular lobe of the kidney, that particular segment of the kidney, and that particular segment will suffer from deficiency of the blood supply. That is one of the specifics of the blood supply of the kidneys. The segmental arteries will be subdivided into lobar arteries, and these continue into the interlobar arteries that we can see here in this picture. So that artery which I am showing you here, that is the interlobar artery. It flows, it goes from the center, from the hilum, from the base, from the apex of the pyramid towards the base of the pyramids. It means that towards the cortex of the kidney. At the junction between the medulla and the cortex, the interlobar arteries branch into the arcuate arteries, as we see here. The arcuate arteries located at the boundary between the cortex and medulla, and from them arise interlobular arteries, which go toward the cortex. From the interlobular arteries arise afferent arteries, afferent arterioles that carry blood into the glomerulus of the kidney corpuscles. Inside of the corpuscle, as we know, there are formed capillary network which compose glomerulus, and from the glomerulus arise efferent arterioles. Efferent arterioles, these are also arterioles, so afferent and efferent arterioles will supply and drain the glomerulus. From efferent arterioles arise capillaries. They will again branch, pay attention here, interlobular artery, afferent arteriole, which carry blood into the glomerulus. Then from them arise efferent arterioles, and then they again will branch into the capillary network. And this capillary network is called peritubular cortical network, which surround the tubules of the nephrons. There's a proximal and distal tubules and so on. Those tubules are surrounded by this peritubular capillary network. And by the way, the endothelial cells of these capillaries produce erythropoietin. Erythropoietin, that is a cytokine, that is a specific substance which will stimulate production of the erythrocytes in the bone marrow. Then from the peritubular network, the blood becomes, gradually it becomes venous blood, and the venous blood via these venules will flow toward the interlobular venules. And so the blood will be drained into the interlobular venules. The interlobular venules flow into the arcuate veins that continue into the interlobar veins, and interlobar veins together, they will join each other and they form the renal veins, which carry blood out from the organ into the vena cava inferior. In this picture, I would like you to pay attention to something else. Here we are looking at from the superficial parts of the interlobular artery, there arise also vessels which will supply the capsule of the organ. So the vessels, arteries also will arise and go toward the cortex to supply the cortex. And from them there will be formed veins which will flow into stellate veins. that they are tributaries also of the interlobular veins. So blood from stellate veins also flow into the interlobular veins. By that way, we told about the blood supply of the cortical nephrons, but we have also blood supply of the juxtamedullary nephrons, most part of which are located at the junction between the cortex and medulla. In this case, the flow of blood till the interlobular arterioles, these are the same as it was for the cortical nephrons. But in this case, there is some difference. Here we see that from the interlobular arteriole arise afferent arteriole, which supply the blood into the glomerulus. From the glomerulus arise efferent arteriole, but in this case, from the efferent arteriole, pay attention, efferent arteriole itself will descend towards the medulla. And here it will form arteriole recti. Arteriole recti, as we see here, and then the arteriole recti descend straight towards the deep layers of the medulla. Then it form hairpin turn, and then it continue into the venule recti, as we see here, venule recti. They ascend towards the corticomedullary junction, and then they will join the arcuate veins. So they carry blood into the arcuate veins. So the arteriole recti and venule recti together, they called vasa recta. This vasa recta is necessary for concentration of the urine, the process which is taking place in the medulla. Also, some branches of the efferent arterioles which arise from the glomeruli of the juxtamedullary nephrons, some of them form network, capillary network, which is peritubular network, around the tubules of the juxtamedullary nephrons. And as it was in the case of cortical nephrons, the blood from here will be formed, will be drained into the venule, which join the interlobular venules, and so on into the arcuate, then interlobar veins. That is a blood supply of the kidneys that we have to know. Now that is here we are looking at the slide of the kidney at the cortex, and here we are looking at the one corpuscle, renal corpuscle in the cross section. Here we can distinguish the capillaries of the glomerulus, and around it we can see the light halo, it's light stripe which surround it, that represented by the capsule, Bowman's capsule, which together form renal corpuscle actually. Then we are looking at the cross sections of the proximal tubules, then distal tubules, and also the medullary, which include collecting tubules. There may be present also sections of arteriole recti, venule recti, and also straight thin tubules of the nephrons. In this picture, we are looking at the apex of the pyramid, the region of the area cribrosa, which have so much openings, as in this case also we see these are actually openings of the collecting tubules, which open onto the at the apex of the pyramid into the minor calyx. In this picture we can distinguish two kidney lobes, one lobe and the other one, and we know that each lobe is represented by the one pyramid, that is the apex of the pyramid, the minor calyx, and the cortex, associated part of the cortex. that is the histological slide also of the one lobule of the kidney, which include one medullary ray and all of the surrounding nephrons, which will be drained into the collecting tubule in the center of this lobule, in particular into the medullary ray. So the nephrons, tubules of nephrons, together with the collecting tubules, they called uriniferous tubules of the kidneys. Here we are looking at the renal corpuscle with all of its structures which are present inside of it. That is the glomerulus, capillaries of the glomerulus, but we cannot see their walls because we know that each corpuscle all include not just capillaries but also capsule. That is a Bowman's capsule. The Bowman's capsule has a parietal layer, as we see here. It is a parietal layer which include which is lined by simple squamous epithelium, and it has also visceral layer. Visceral layer is formed by podocytes that surround the capillaries. Almost all parts of the capillaries are surrounded by the podocytes. But podocytes, they do not, their cell bodies do not touch the capillaries. Actually, from each cell body of the podocyte arise two large processes, cytotrabecules. From the cytotrabecule arise small processes which called cytopodia, as we see here. So we have primary process as this one, and secondary processes they called pedicels. The pedicels and pedicels will touch the basal laminum of the podocytes, and as we know that they surround the capillaries. The capillaries here, of the glomerulus, they belong to fenestrated capillaries which do not have diaphragms. They are devoid of diaphragms. So the endothelial cells, basal lamina, together with the basal lamina of the podocytes, they touch each other and they form basement membrane, which is the one of important parts of the filtration barrier. So between the bloodstream and the lumen of this Bowman's capsule, there is formed the filtration barrier. The filtration barrier is formed by, first of all, endothelial lining of the blood capillaries with no diaphragms, and there are slits, these filtrations, these fenestrae, they stop the passage of substances which are larger than 90 kilodalton. Then we have basement membrane, about which we will talk with you later, and we have also filtration slits. These are slits, these are spaces between the neighbor pedicels. And the filtration slits, here we see these filtration slits which are not open, and they are covered by filtration slit diaphragm. So between the neighbor pedicels we have the diaphragms that include small openings, and they will also stop passage of substances which are larger than 80-90 kilodalton. The main part of the filtration barrier is formed by the basement membrane. We will see the basement membrane on next pictures, but before it, I would like you to pay attention to this filtration slit diaphragm. That is a pedicel, one pedicel, and the neighbor pedicel. That is a glomerular basement membrane, about which I just mentioned, and endothelial cells or the blood capillaries with fenestrae. We can see here that between pedicels we have diaphragm, and this diaphragm has small openings, and this diaphragm is formed by specialized also proteins We can see here p-catechols, alpha-actinin, which is present on the side of the pedicel. Also, there are present nephrin proteins. The genetical mutations of the nephrin protein may lead to very dangerous consequences in the filtration process. So that is a filtration diaphragms, and so the pedicels will envelope our capillaries, as we see here, and the basement membrane here, which is present between the pedicels and the endothelial cells, it has a three-layered structure, and it is the product of in one side of the endothelial cells and on the other side of the pedicels of the podocytes. Here we are looking at the lumen of the blood capillary and the direction of the blood plasma towards the lumen of the Bowman's capsule. So that is the first step in the production of the urine. The podocytes we can see here, the pedicels which attach to the basement membrane, and filtration slits can be distinguished, as you see here, filtration slits. And on the side of the endothelial of the capillaries, we can also distinguish here the fenestrae. These are openings between the endothelial cells, and the formation of the ultrafiltrate. Finally, the basement membrane. The basement membrane, we have to know that it includes three layers. That is the electron micrograph representation of the filtration slits, filtration barrier, that include again the pedicels between the filtration slits, then endothelial cells lining of the blood capillary, and between them the common basement membrane. The basement membrane here has thickness approximately 300 nanometers. It includes three layers, two electron-lucent layers, which called lamina rara, and between them, in the center, located lamina densa. Lamina rare or lucid layers, they include mainly glycoproteins, proteoglycans, perlacans, which reach in heparan sulfate, and heparan sulfate, as we know, it reach in negatively charged ions. So these heparan sulfates will prevent passage of negatively charged ions through the filtration slits. In the center, the lamina densa, it includes the network of collagen 4 type that form network in the lamina densa, and it will prevent passage of substances which have size more than 69 kilodalton. So finally, what will be passed through the filtration barrier, it will be plasma ultrafiltrate that devoid of blood cells and devoid of large proteins, which are larger than 69 kilodalton. In this picture, we are looking also at the podocyte electron micrograph representation, and here the pedicels we can see, and the lumen of the capillary. This one also shows us the capillaries of the capillary network of the glomerulus, and they are surrounded by the pedicels of the podocytes. That is a filtration barrier also with all of its components. The capillary in the center, podocytes, the primary processes and secondary processes we can distinguish here. Also, we have to know that the kidneys, so the Bowman's capsule, it continues into the proximal convoluted tubule that lined by simple cuboidal epithelium. So the plasma ultrafiltrate, which is approximately 180 liters of which is formed during 1 day, then it flows into the tubules of the kidneys or of the nephrons for further steps in the urine production. It will be reabsorption of most of the components which are filtrated through the filtration barrier. This one, in this case, we are looking at the arteriole, afferent arteriole, efferent arteriole. They form here the trigone structure, and to this trigone attached distal convoluted tubule, because they have specialized, they have some purpose for this location. And in this case, we can see differences in the histological structure of different parts of kidney tubules. Actually, in the proximal convoluted tubule, the cells, they are cuboidal, and we cannot distinguish the boundaries between the neighbor cells because there are lots of interdigitations between the cells. And on the apical surface, there is brush border that is formed by lots of microvilli that increase surface area, which is necessary for the reabsorption to take place. In the basal part of the cells, there are also many invaginations. Between them, there are lots of mitochondria. All of these structures are needed for the reabsorption to take place because the most part of ultrafiltrate, which is formed in the renal corpuscle, it is reabsorbed via cells of the proximal tubules into the bloodstream. Inside of the cells also were a well-developed endosomal compartment, which is necessary for the absorption of solutes and large macromolecules from the lumen of the proximal tubules. Then straight tubules, they descending limb, ascending limb, they are covered by simple squamous epithelium. A descending limb, there are cells will absorb the water and salts, but in ascending limb, the epithelium is impermeable for the water, and there will take place absorption of mainly salts, sodium, chloride. And in distal straight tubule, then in distal convoluted tubules, there also will take place absorption of the salts because the cells of these regions, they are rich in the sodium chloride and potassium transporters. Also, sodium potassium pumps in the basal part of the cells that they will actively resorb the sodium and chloride from the lumen of the tubule. Finally, from the distal tubule, the urine flows into the collecting tubules. In the collecting tube, also in distal tubules, the distal tubules are covered by simple cuboidal epithelium. But here the amount of microvilli, also interdigitations, they are less than it was in proximal tubules. Also, length of proximal tubules approximately 7 times longer than length of the distal tubules. That's why in the cross sections, in the slides of the kidneys, mainly we will see proximal tubules. The ratio there will be 7:1, in which 7 the proximal tubules and 1 it is the distal tubules. In collecting tubules, they are covered by simple cuboidal to columnar epithelium. Here we will present two kinds of cells. They are the principal cells and the intercalated cells. The principal cells, they have receptors for aquaporin, for the antidiuretic hormone, ADH. In the response to ADH, the cells will in the cells will be taking place absorption of the water from the final urine. And intercalated cells, they reach in potassium pumps, and the transporters of potassium with NH4 that is necessary for releasing of H protons into the lumen of the tubule, and by that way they will participate in the acidification of the final urine. So the final urine will be acid, will be hyperosmotic, and it contains less amount of the fluid. In this case, we can see the cells of the proximal tubule. We cannot distinguish the boundaries between because so much interdigitations, basal foldings. In apical part, there are lots of microvilli which form the brush border. All of them are necessary for proper reabsorption of the necessary materials. Electron micrograph representation also shows us the structure of the cells in the proximal tubules. These are brush border microvilli and invaginations, basal invaginations, and between the mitochondria we can distinguish in the electron micrograph. That is the in the distal tubule, and that they contain less amount of microvilli on the surface and less amount of infoldings, basal infoldings. So we can see here that when how the process of reabsorption is taking place. Actually, in proximal convoluted tubule, most part of the water, sodium, chloride, and HCO3 hydrocarbonate anions, they are absorbed in the proximal tubules. Approximately 67 to 8% of these substances are absorbed in the proximal tubules. Also, 100% of proteins, amino acids, glucose, vitamins, they absorbed in proximal tubules. By that way, when also most of the creatinine is absorbed, so when the urine passes from the proximal tubule, its osmolar activity, its osmolarity will not change. It will be equal to that of the blood plasma. So it will equal to 300 milliosmos. But when the urine flows into the distal tubule towards the thin tubules, in thin tubules we will see absorption of the water and also salts, but more water. That's why the urine inside of distal tubules will undergo concentration, and finally when it will reach the loop of Henle, the osmolarity inside of the tubule will reach 1200 milliosmos, in opposite to 300 which was in proximal tubule. Then it flows towards the distal tubule, and in distal tubule the epithelial cells are impermeable for water, but it is permeable for sodium chloride ions, and that's why when the urine flows towards the distal straight tubule, towards the distal convoluted tubule, the urine, because it loses salts, its osmolarity again drops from 1200 to 300, and that urine, which will osmolarity of which will be equalized with the plasma, which will be 300, it flows into the distal convoluted tubule. So in distal straight tubules there are sodium, potassium pumps, and the process of active reabsorption of sodium and chloride ions will lead to the decreasing of the concentration of the urine. Then that urine, as we see here, it flows towards the distal convoluted tubules, and in these tubules there is the process will absorption of the reabsorption will be controlled by the hormones. It mainly aldosterone. Aldosterone, the hormone which will control absorption of the sodium ions, will increase absorption of the sodium ions. Also, we have parathormone, which will stimulate reabsorption of the calcium salts, and so that urine will flow into the collecting tubules. In collecting tubules, the epithelial cells are impermeable for water, but because of the activity of the ADH hormone, the ADH will stimulate cells of the collecting tubules to resorb the water. That's why the osmolarity of the urine will gradually increase towards the medulla, towards the apex of the pyramid. But in the deep layers of the medulla, the process will also influenced by the hyperosmotic pressure of the interstitial tissue of the kidney, which has 1200 milliosmos. That's why the osmolarity of the urine will be equalized to the osmolarity of the interstitial tissue, and it will reach 1200 milliosmos when it will come towards the papilla of the renal pyramid. So in this picture we can see that the process of reabsorption, it is complex process. It is influenced by many factors. In the proximal tubules, we are seeing the process of obligate reabsorption. But in distal parts, we can see the absorption which is optional. So in the first part, it is vital, the reabsorption, secretion of lactates, ketones, and other urea also will be released. But in the distal tubules, we can see optional, which depends on the activity of the hormones, which depends on the demands of the body. And ADH will activate the absorption of the water. As we see here, that is a collecting tubule, that is the principal cell of the collecting tubule. And in the basal part, it has receptor for ADH hormone. ADH hormone from the blood plasma will come and attach to its receptor, and on the response to that hormone, the cells will release their aquaporin channels into the cell membrane. Aquaporin 2 will be incorporated into the cell membrane and will stimulate, will provide absorption of the water. So the water through these water channels will enter the cell, and in the basal part we have aquaporin 3, through which water will enter first of all into the connective tissue, into the interstitial tissue, and then into the lumen of the blood vessels. So the absorption of the water will take place. So in the problems of deficiency of the ADH hormone or of its receptor or of the aquaporin channels, the absorption of the water will be interrupted, will be disturbed, and the body will lose large amounts of the fluid. So we know that kidneys, by elimination of the excess of toxins, excess of fluid, it also participates in the regulation of the water-base balance, fluid balance of the body. In the basal part of these cells of the, for example, kidney tubules, we have sodium-potassium pumps. Here we can see the mechanism of absorption. In the basal part, we have sodium-potassium pump through which 3 sodiums go out of the cell and 2 potassiums enter the cell. So the sodium-potassium pump keep low levels, low concentrations of the sodium inside of the cell, and in response to it, In the apical part, we have the transporters of sodium potassium chloride, which will carry these ions into the cell. Then through the basal membrane, chloride and sodium will be released into the interstitial tissue and through it into the bloodstream. So the reabsorption is taking place in the tubules. Actually, the juxtaglomerular nephrons, together with the vasa recta, they form the countercurrent exchange system inside of the kidneys. There are countercurrent exchange system, and we also have countercurrent multiplier system. Actually, the vasa recta, they form exchange system, and the countercurrent flow system will be formed by the juxtamedullary nephrons. They will return the water and salts into the bloodstream, because when the artery recti and vena recti, one of them descend, other one ascend inside of the medulla, they will gain the water and salts from the interstitial tissue of the kidneys, and more water, more solutes will be drained out into the venous system than they are filtrated through the wall of the arteriole recti, because of the narrow lumen of the arteriole recti. Then we can see here the endocrine system of the kidney, endocrine apparatus of the kidney. By other way, it is called juxtaglomerular apparatus. Here we can see juxtaglomerular cells. These are the smooth muscle cells in the wall of the arteriole recti. They are cuboidal in shape, and they contain granules, secretory granules with renin substance, because these cells produce renin. Also inside of the cytoplasm, you can find the angiotensin converting enzyme, angiotensin 1 and 2. Then the macula densa, this is the epithelial lining of the distal convoluted tubule, which attaches to this trigone region. That is that is a vascular pole of the corpuscle, and that is a urinary pole. In vascular pole, we are looking at the arteriole recti, then that is afferent arteriole, then efferent arteriole, and between them the distal convoluted tubule attached to this region, and the epithelial lining of this tubule at this region becomes highly tall, columnar. They were close to each other. That's why they form dense line. That's why the name of this structure. And under these cells, we cannot see the basal lamina. Inside of the cytoplasm of these cells, there is the nitric oxide synthase. Also, there is present the enzyme SOX2, which is needed for production of the prostaglandins. Here we can see the mesangial cells. Mesangial cells, some of them located outside of glomerulus, that's why they called extraglomerular mesangial cells, and some of them located inside of the glomerulus, between the capillaries of glomerulus. These are intraglomerular mesangial cells. The mesangial cells, they actually the supporting cells. They phagocytose the spent part of the basement membrane, and they also participate in the production of the basement membrane. Then they have receptors for angiotensin II, in response to which they contract and they decrease filtration. By that way, they decrease production of the urine, decrease filtration of the blood plasma. And also here the extraglomerular mesangial cells, which located at the trigone between the afferent and efferent arteriole, they may provide functions of the juxtaglomerular cells if they are disturbed. The macula densa actually, the cells, they are chemoreceptors which will sense a concentration of the sodium ions inside of the distal convoluted tubule. And if the amount of sodium ions decrease inside of the urine, final urine, which flows through the distal tubule, the cells will receive that information, and in response to it, the sodium oxide synthase is activated. It produces nitric oxide synthase is activated. It produces nitric oxide, and nitric oxide via diffusion will reach the juxtaglomerular cells, and they will produce renin in response to decreasing amount concentration of the sodium ions inside of the distal tubule. Also, these cells produce prostaglandins. They also will pass and reach these juxtaglomerular cells and will also stimulate the production of the renin. What will do renin we will see on next figures. But here you are looking at the podocytes, their processes, primary, secondary processes, and also juxtaglomerular, the mesangial cells here. And that is a slide of which represents the endocrine apparatus of the kidneys with the macula densa, then the vascular pool, and all of the surrounding structures. The production of the renin in response to decreasing of concentration of sodium ions, or renin also may be produced in response to decrease in renal perfusion. So if the blood pressure in afferent arteriole will drop, in response to it also may be production of the renin. The renin will assist in converting the angiotensinogen, which is produced in the liver, into angiotensin first. Angiotensin first, under the influence of angiotensin converting enzyme, which is present in the lung capillaries, will be converted into angiotensin second, and angiotensin second will activate sympathetic activity, will increase absorption of the sodium and chloride from the kidney tubules, will stimulate adrenal gland cortex of the adrenal gland to produce aldosterone. Aldosterone in turn will also stimulate absorption of the sodium ions in distal convoluted tubules. Then angiotensin second also stimulates the production of the ADH from the neurohypophysis, and so it will increase absorption of the water. So finally, the activity of angiotensin second will lead to increase of the blood volume, increase of the water, increase of the level of the sodium ions inside of the body. All of them will lead to increase of arterial pressure. Also, the vasoconstriction. So all of them will lead to the increase of the arterial pressure in our body. The excretor passage of the urinary system that start from the renal calyces and finalized in urethra, all of them are covered, all of these organs are covered by transitional epithelium. It is, as we know, it is a type of stratified epithelium, and it includes several layers of cells. The transitional epithelium will change its thickness according to the state of the organ. And if the organ is full, the epithelium will flatten, and because it will stretch. But if the organ is empty, it will have several layers of cells, and even the superficial layer will have dome-shaped cells. Some of them may have two nuclei. Under the epithelium, we have lamina propria, which include, that is a connective tissue with the blood vessels, with nerve supply, nerve endings, and under that is a tunica mucosa. Under the mucosa, we will have the muscularis, which include in ureter, it is inner longitudinal and outer circular layers. But in urinary bladder, there we will see 3 layers, inner and outer longitudinal and middle circular layers. What about the cells of the superficial layer of the transitional epithelium? As we see here, the membrane, the cell membrane of the apical part of these cells, it has some plaque regions and interplaque regions. The plaque regions include its specialized content, which is necessary, first of all, for formation of the foldings, hinge regions, as we see here, and which is necessary for stretching out the surface of the membrane. And otherwise it's on other hand, it necessary for protection of the cell membrane against hyperosmolar urine, which is present in the lumen of the urinary bladder. That is storage of the urine, and that's so because of the high osmolarity, it may have dangerous effect on the cells. That's why the cell membrane will have its own content with the plaques and interplaque regions. So that is the about the urinary system. And now here we are looking at the development of the urinary system, because during the development we have 3 stages. It will be pronephros, mesonephros, and metanephros. The development of the urinary system, it starts at the beginning of the 4th week, and on which we will have pronephros, that is the segmented intermediate mesoderm, which represented by the nephrogonotomes, which will present in the neck region of the embryo. Then we will have mesonephros, which starts at the low neck, and then thoracic segments, which also represented by the nephrogonotomes, segmented nephrogonotomes, and then unsegmented nephrogenic cord will also participate in the formation of the mesonephros. And finally, we will have metanephros. It will be definitive kidneys, which develop from the metanephrogenic blastema, and ureters, which will grow as outgrowths of the cloaca, that will form ureteric bud, which grow into the metanephric blastema. From metanephric blastema will grow the kidney tubules, the nephrons, the tubules of nephrons, but the collecting tubules, then calyces of the kidneys, the ureters, all of them will take origin from the ureteric bud. Then they will join each other, so they have different origins for development. Then the female reproductive system. As we know, the female reproductive system, it represented by the paired ovary, then paired uterine tubes, fallopian tubes, uterus, vagina, and external genitalia. The main glands which produce female sexual gland hormones and female sexual cells, these are ovary. Actually, ovary, then uterine tubes, they belong to intraperitoneal organs. It means that they are covered by peritoneum in all their sides. If we will look at the ovarium structure, we can see that it's covered by the germinative epithelium, under of which we have tunica albuginea. It is a dense connective tissue coat that surrounds the ovarium. Then under of it we have cortex and the medulla. In cortex of the ovarium we have follicles in different stages of development. Their development starts from the primordial follicle. Then in each menstrual cycle, approximately 20 of primordial follicles start their growth from the start of puberty time, and in each menstrual cycle 20 of these follicles start their growth till they reach the fully mature follicle. Only one of them may reach that fully mature state. Others are dying by atresia. When the growth of follicles is taking place, they will go from primordial to primary, then there will be formed secondary, tertiary follicles, and tertiary follicle will, when it will reach 14th day of menstrual cycle, it is a midline. The follicle will undergo ovulation, and the oocyte together with its surrounding coats will be released from the ovarium and enter the uterine tube. As we know, during the ovulation, because the oocyte is going out from the follicle, then in the place of the follicle will be formed, first of all, corpus hemorrhagicum, as the structural integrity of the blood vessels of the wall of the follicle is interrupted. The leakage of the blood from those blood vessels will lead to accumulation of the blood inside of the lumen of the ovulated follicle. It will lead to formation of the corpus hemorrhagicum. Then the cells of the follicle will grow into the actively hormone producing cells, and they will accumulate the yellow pigment, and there will form the corpus luteum. It is the yellow body which produces necessary hormones in the second part of the menstrual cycle. The yellow body may be called the yellow body of the menstruation, or if the pregnancy will take place, will happen on that case under the influence of the hormones chorionic gonadotropin that is produced by placenta. The yellow body will continue its functioning till the 5th month of pregnancy, and on that case the yellow body is called the yellow body of pregnancy, and it is necessary to produce progesterone, estrogens, the hormones that are needed for the maintenance of the pregnancy. When the yellow body ceases its functioning, when it will finish its functioning, it will be converted into the white body. It is a scar tissue that the hormone producing cells will die by necrosis, and they will be replaced by connective tissue cells that they will form scar tissue. So in the cortex we have all of these structures. We can see all of these structures, but the medulla of the ovarium is not well developed. There is just a little bit of interstitial tissue. There may be present interstitial glands that produce a little bit of estrogens or androgens. So the medulla in the ovary, it's not well developed. And if we look at the slides of the ovarium, we can see all of the structures. As we know, the primordial follicles, these are large, vast majority of the follicles which are located near to the capsule, near to the tunica albuginea, and under of it we have follicles in different stages of development. We can find here primary follicle, we can find here secondary or tertiary follicles, which are in different stages of the development, and interstitial tissue that is present between them we can also distinguish here. How can we distinguish the primordial follicle from the primary and so on? Actually, we have to remember that the embryology material which we studied, we know that process of oogenesis, it starts at the embryonic life of the at approximately 6 to 7 weeks of gestation, and you know that primary sexual cells they develop inside of the wall of the yolk sac. Then they migrate into the body of the embryo, and they migrate towards the gonads, which are indifferent, and which are developed in the midline of the mesonephros. The genome of the primary sexual cells will determine the further pathway for the development of gonads, because initially gonads, they are just indifferent, they are masses of the intermediate mesoderm which formed in the both parts of the midline. But when the primary sexual cells populate the gonads, they will, under the influence of the genome, the gonads will determine the pathway. They will develop towards a male or female reproductive system. If the genome will contain Y chromosome, the Y chromosome, sexual chromosome, it contains the sex determining factor region that will determine the male embryo, fate of the male embryo. But if there are no Y chromosome, so the gonads will develop towards the ovary. And inside of the ovary, so the ovary will develop as it's needed, and the ovogonia, which are the primary sexual cells, they undergo division, and there are formed 7 millions of ovogonia. From 7 millions, 1 to 2 millions, then they will be incorporated into the follicular cells, and they will grow and will form primary oocytes. So the primary oocyte with surrounding follicular cells, they form primordial follicles. From the primordial follicles will grow the primary follicles during the puberty. But till puberty, the follicles will remain at this stage, at the primordial follicle stage. To the birth time, inside of the ovary of the female embryo, female newborn child, in her ovary will be approximately 1 to 2 millions of primordial follicles. From them to the time of puberty will survive just 350 to 400,000 of primordial follicles, and from them just 350 of them will fully develop during all sexual active period of the life of the female. During each menstrual cycle, so 20 approximately of primordial follicles will start their growth, and the primordial follicles will be converted into the primary follicle. In primary follicle, we will see the formation of the zona pellucida around the oocyte. If the zona pellucida is formed, it means that that is the primary follicle. It may have one layer of follicular cells or several layers. It will with one layer or multi-layered follicular cells around the oocyte in primary follicle. Then the primary follicle will grow into the secondary follicle. In secondary follicle, between the follicular cells or granulosa cells, they called, between them we will have spaces which will be filled with the fluid, tissue fluid, follicular fluid. And when they will converge with each other, there are formed one cavity with the follicular fluid, and it will be follicular cavity. Antrum is called. Oocyte, together with its surrounding granulosa cells, then zona pellucida, will be located in one pool. That is the in Graafian, it is a tertiary follicle which will undergo ovulation. So then the uterus is the next organ. Here you can see the layers of the human uterus. Uterus is a muscular organ, is necessary for development of the fetus. And there we have endometrium, it is tunica mucosa, then myometrium, next coat, and the perimetrium, it is the outer coat of the uterus. In number 1 here we can see that's a large magnification of the one part of this picture, histological slide stained by hematoxylin eosin, and here we are looking at the endometrium, the tunica mucosa, then myometrium, which have three layers, inner and outer longitudinal layers of muscles, and middle circular layer. Actually, in middle circular layer, we have blood vessels. That's why this layer is called vascular layer. And outer is perimetrium, which is covered by mesothelium, that is a tunica serosa. Here we are looking at the stage of the menstrual cycle and what is taking place in the ovary. In ovary, the stages of menstrual cycle are called. First stage, it's follicular stage, when the growth of follicles is taking place. Then ovulation we will have, and then lutein stage, the period of growth of the yellow body. But in uterus, as you see here, that is the endometrium. In the first stage, we will have menstruation. Then will be proliferative stage, that is characterized by proliferation of the endometrium, of the layers of the endometrium. And then the second stage will be called secretor stage. It is characterized by spiraling of the glands of the endometrium. Then the blood vessels will grow, that is preparing the wall of the uterus for the implantation to take place. If the implantation will not take place, as we know, the endometrium include basal layer and functional layer. The functional layer of endometrium will go off during the menstruation, during 5 days, and the functional layer will detach out from the surface of the endometrium, and the basal layer will remain and serve as regenerative pool for the regeneration, for the proliferation of the endometrium, functional layer of endometrium on the next menstrual cycle. That's the level of hormones that will also change according to stages of menstrual cycle. So in the first stage of menstrual cycle, we will have increasing the level of FSH, follicular stimulating hormone, that is produced by hypophysis, then increasing of the level of estrogens, and then near to the 14th day of menstrual cycle, near to the ovulation time, these levels will drop, but LH, luteinizing hormone, will increase. The number 11 indicate the level of luteinizing hormone. Then it will drop on the next stage, in second stage of the menstrual cycle. But in second stage, we will see increasing surge of the level of progesterone, which is produced by the yellow body, which prepares the endometrium for the implantation. Also, estrogens will again surge for, it also will be needed for the cyclic change that take place in the wall of the endometrium. So, and the temperature of the uterus, the intravaginal temperature will be surged in the midline of the menstrual cycle. It will reach approximately to the 37 grade, but in other times it will be approximately 36 and a little bit more than 36 grade by Celsius. Uterine tubes or fallopian tubes, the cross section here we are looking, that's covered by tunica mucosa, then muscularis, and outer one will be serous layer. Here the uterine tubes, like in uterus, the epithelium will be simple columnar. There will be present two kinds of cells, ciliated cells and PEG cells or secretor cells that produce mucus, that secrete into the lumen of the tube in the case of the fallopian tube, and if it is in the uterus, it will be produced into the lumen of the uterus. Under of epithelium, we will have tunica, we will have lamina propria with blood vessels, with all of the necessary components. The cyclic change, again, we see here, that's just I mentioned. And so we are coming to the male reproductive system. The male reproductive system, here the glands, which are paired glands, which are analogous of the ovary in the female reproductive system, they are testes. The testes, they are located in scrotum, paired organs. They participate in the production of the male sexual hormones, testosterone and dihydrotestosterone, and also production of the male sexual cells. The structure of the testis we can characterize as it has lots of coats actually. The first coat, it is a tunica vaginalis. Then we have tunica albuginea, which is the actually tunica vaginalis, it is part of peritoneum, but albuginea is dense irregular connective tissue which form the capsule for this organ. And in the back part of the organ, it forms thickening which is called mediastinum testis. From mediastinum testis, there are septa, lots of septa trabecules which enter the organ and subdivide it into the individual lobules. Each lobule has pyramidal shape. There are approximately 250 lobules in each testis. Inside of each lobule, we have 3 to 4 seminiferous tubules. Seminiferous tubules, these are main components of the testis which produce spermatozoa, the male sexual cells. Then behind each testis we have the additional part which attaches to the testis, and it is the epididymis actually, that is epididymis. If we will characterize the parts of the male reproductive system, we should mention here the lots of seminiferous ducts which carry the semen. Then there are paired additional glands which may be paired or unpaired. Paired glands, these are seminal vesicles. Pay attention, in this case here the seminal vesicles, then bulbourethral glands, and unpaired prostate gland. So here we can see the paired testis, then paired seminal vesicles and bulbourethral glands. But unpaired, it is the prostate gland as we see here, and lots of duct systems we can see here. And and penis, that's also a part of the male reproductive system. If we are talking about the internal structure of the testis, as we see here the lobules of the testis, seminiferous tubules which arise inside of the testis, they continue into the tubuli recti. These are straight tubules that join the rete testis, which is inside of the mediastinum testis. From the mediastinum testis arise ductuli efferents, efferent ductules, approximately 15 in amount. Then they join the ductus epididymis, which is highly coiled, and it forms the head, then body, and the tail of the epididymis. From the tail epididymis arise ductus deferens, that is ductus deferens that enters the body, as you see here, ductus deferens, that goes behind the urinary bladder, and there it continues and it will join the ductus of the seminal vesicles at the level of the prostate gland, and they will join each other and they will form the ejaculatory duct. Ejaculatory duct is joining the male urethra. So male urethra has double function, not just releasing of the urine, but also it will serve as duct for the genital system for the releasing of the semen. So that's the general structure of the male reproductive system. In the histological slide, we can see the testis with the surface of the testis with tunica vaginalis. Then we have the head of the epididymis, which are located very close to each other actually. If we are looking at the seminiferous tubule structure, we can see here the mainly two kinds of cells in seminiferous epithelium, which covers the seminiferous tubule. We can distinguish mainly two kinds of cells. First of all, these are Sertoli cells. These are supporting cells, and their lateral processes join each other via tight junctions, and they form hematotesticular barrier here, which is necessary for protection of the developing spermatozoa from the immune system of the body. So the Sertoli cells subdivide the seminiferous epithelium into two compartments. That is basal compartment, which is below, and there are located spermatogonia, and above compartment, which is called abluminal compartment. And here we're located the all other cells, beginning from the primary spermatocytes till the spermatozoa. Under of epithelium, we have basal lamina, then tunica, there will be lamina propria of mucosa that include myoid cells, which have contractile ability. The process of spermatogenesis we can see here that starts from the spermatogonia, and there are spermatogonia dark type A, type B, then they converted into spermatogonia B. The spermatogonia B will enter the growth stage, and they form primary spermatocytes. Then they enter meiosis, first division of meiosis, and they form secondary spermatocytes. And the secondary spermatocytes will again divide by meiosis 2, and they form early spermatids. Then spermatids, because they have rounded shape, they should undergo spermiogenesis, during which they will change their shape. They undergo some morphological change, and they will form, finally there are formed mature spermatozoa. All of these processes, of course, under the hormonal control. As we know, hypothalamus produces gonadoliberins. These are LH releasing factor or luteinizating hormone releasing factor, or FSH releasing factor. Under the influence of hypothalamic hormones, the hypophysis will produce FSH or LH. For FSH, the Sertoli cells have receptors, and under the influence of FSH, Sertoli cells produce androgen binding protein, which is necessary to bind testosterone. But we know that besides them, in the interstitial tissue of the testis around the seminiferous tubules, we have interstitial cells, which they called Leydig cells, and they produce testosterone. Leydig cells these are interstitial cells. They have receptors for LH, for luteinizating hormone. Under of influence of LH, these cells produce testosterone. But to be able to that testosterone to reach spermatogenic epithelia, to reach developing spermatozoa, there is needed androgen binding protein, which is produced by the Sertoli cells. So Sertoli cells will bind that, will produce that protein, which will bind testosterone and carry it towards the spermatogenic epithelium. So here we can see the prostate gland, in the center of which we have male urethra. Male urethra has three parts actually. That is prostate part, then which passes through the center of the prostate gland. We have also membranous part, which passes through the perineum, so the diaphragm. And we have also the final part, it will be spongious part, which is located inside of the penis. Most of the parts of the male urethra, the length of male urethra is 20 centimeters, but in female it is 5 centimeters. Most of its parts is covered by transitional epithelium, but starting from the perineal part, from the membranous part, and all of the spongious part is covered by stratified columnar epithelium. Near to the glans penis, it will be covered by stratified squamous epithelium. Prostate gland, it is a complex of glands which subdivided into 3 regions. It is mucous glands, submucous glands, and the main prostatic glands, which are located in most periphery. The ducts of them, all of them open into the male urethra. Each of the glands inside of the male reproductive system, it produces its own secret. Actually, the 70% of the semen is produced by the seminal vesicles. The main component of the secretion of the seminal vesicles, it is the fructose, which is necessary for nourishment of spermatozoa. Prostate gland also produces its own secret, which is rich in prostaglandins, in acid phosphatases, in citric acid, and so neutral proteases. They will be released content. Then the bulbourethral glands produce the secret at the final, in the last stage, finally, and they mainly produce mucus that will lubricate the surface of the male urethra. So that's about the development. That's the structure of the male reproductive system. So that is the end of the lecture. Thank you for your attention.